Principles of inheritance and variation. Have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animals? Or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offsprings identical to their parents, or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other, or sometimes even so different? These and several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance, as well as the variation of characters from parents to offspring. Inheritance is the process by which characters are passed on from parent to progeny. It is the basis of heredity. Variation is the degree by which progeny differ from their parents. Humans knew from as early as 8000 to 1000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variation that were naturally present in the wild populations of plants and animals to selectively breed and select for organisms that possessed desirable characters. For example, through artificial selection and domestication from ancestral wild cows, we have well-known Indian breeds. Example, Sahiwal cows in Punjab. We must, however, recognize that though our ancestors knew about the inheritance of characters and variation, they had very little idea about the scientific basis of these phenomena. 5.1 Mendel's Law of Inheritance. It was during the mid-19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiment on garden peas for 7 years. It is from 1856 to 1863 and proposed the laws of inheritance in living organisms. During Mendel's investigation into inheritance patterns, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiments had a large sampling size, which gave greater credibility to the data that he collected. Also, the confirmation of his inference from experiments on successive generations of his test plants proved that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposite traits, example tall or dark plant, yellow or green seeds. This allowed him to set up basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observations and the complexity inherent in them. Mendel conducted such artificial pollination or cross-pollination experiments using several true breeding pea lines. A true breeding line is one that, having undergone continuous self-pollination, shows the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated, full or constricted green or yellow pods, and tall or dark plants. Table 5.1 Contrasting traits studied by Mendel. Characters and contrasting traits. Stem height, that is tall and dark. Flower color, that is violet and white. Flower position, that is axial and terminal. Pod shape, that is inflated and constricted. Pod color, that is green and yellow. And seed shape, that is round or wrinkled, and seed color, that is yellow and green. 5.2 Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel where he crossed tall and dark pea plants to study their inheritance of one gene. He collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. This generation is also called the filial progeny or the F1. Mendel observed that all the F1 progeny plants were tall, like one of its parents. None were dark. He made similar observation for the other pairs of traits. He found that the F1 always resembled either one of the parents and that the trait of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants and to his surprise found that in the filial 2 generation, some of the offspring were dark. The characters that was not seen in the F1 generation was now expressed. The proportion of plants that were dark were 1/4 of the F2 plants while 3/4 of the F2 plants were tall. The tall and dark traits were identical to their parental type and did not show any blending. That is, all the offspring were either tall or dark. None were of in-between height. Similar results were obtained with the other traits that he studied. Only one of the parental trait was expressed in the F1 generation while at the F2 stage both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending at either F1 or F2 stage. Based on these observations, Mendel proposed that something was being stably passed down, unchanged from parent to offspring through the gametes over successive generation. He called these things as factors. Now we call them as genes. Genes therefore are the unit of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. That is, they are slightly different forms of the same gene. If we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the character of high, T is used for the tall trait and small t for the dark. And capital T and small t are alleles of each other. Hence, in plants, the pair of alleles of height would be capital T, capital T and small t, or small t, small t. Mendel also proposed that in a true breeding, tall or dwarf pea variety, the allelic pair of genes for height are identical or homozygous, capital TT and small tt. Respectively, capital TT and small tt are called the genotype of the plant, while the descriptive terms tall and dwarf are the phenotype. What then would be the phenotype of a plant that had a genotype capital T and small t? As Mendel found the phenotype of the F1 heterozygote, that is capital T small t, to be exactly like the capital T parent in appearance, he proposed that in a pair of dissimilar factors, one dominates the other, and hence is called the dominant factor while the other factor is recessive. In this case, capital T is dominant over small t, that is for the dwarfness, that is recessive. He observed identical behavior for all the other characters, that is trait or pairs, that he studied. It is convenient or logical to use the capital and lower case of an alphabetical symbol to remember this concept of dominance and recessiveness. Do not use capital T for tall and D for dwarf, because you will find it difficult to remember whether capital D and small d are alleles of the same gene or character or not. Alleles can be similar as in the case of homozygotes, that is capital T and small t, or can be dissimilar as in the case of heterozygote, that is capital T and small t. Since the capital T small t plant is heterozygous for gene controlling one character, that is height, it is monohybrid, and the cross between capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair separate or segregate from each other and only one allele is transmitted to a gamete. This generation of alleles is a random process, and so there is a 50% chance of gamete containing either allele, as has been verified by the results of the crossing. In this way, the gametes of the tall TT plants, capital TT plants, have the allele capital T, and the gametes of the dwarf small TT plants have the allele small t. During fertilization, the two alleles, capital T from one parent, say through the pollen, and small t from the other parent, then through the egg, are united to produce zygote that have one capital T allele and one small t allele. In other words, the hybrids have capital T small t since these hybrids contain alleles which express contrasting traits. The plants are heterozygous. The production of gametes by the parents, the formation of zygotes, the F1 and F2 plants can be as shown in figure 5.4. It was developed by a British geneticist, Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible gametes are written on two sides, usually the top row and the left columns. All possible combinations are represented in boxes below in the squares, which generates a square output form. The Punnett square shows the parental tall capital T male and dwarf small t female plants, the gametes produced by them, and the F1 capital T small t progeny. The F1 plants of genotype capital T small t are self-pollinated. The symbols male and female are used to denote the female eggs and male pollen of the F1 generation respectively. The F1 plant of the genotype capital T small t, when self-pollinated, produces gametes of the genotype capital T and small t in equal proportion. When fertilization takes place, the pollen grains of genotype capital T have a 50% chance to pollinate eggs of the genotype capital T as well as of genotype small t. Also, pollen grains of genotype small t have a 50% chance of pollinating eggs of genotype capital T as well as of genotype small t. As a result of random fertilization, the resultant zygotes can be of the genotypes capital T small t, or small t small t. From the Punnett square, it is easily seen that 1/4 of the random fertilization lead to capital T, 1/2 lead to heterozygous, and 1/4 to the recessive. Though the F1 have a genotype of heterozygous, but the phenotypic character seen is tall at F2. 3/4 of the tall plants, where some of them are capital T, while others are capital T small t. Externally, it is not possible to distinguish between the plants with the genotypes dominant one and heterozygous. Hence, within the genotypic pair heterozygous, only one character tall is expressed. Hence, the character capital T or tall is said to be dominant over the other allele small t or dwarf character. It is thus due to this dominance of one character over the other that all the F1 are tall, and in the F2, 3/4 of the plants are tall. This leads to a phenotypic ratio of 3/4 tall and 1/4 dwarf, that is 3:1 ratio, but a genotypic ratio of 1:2:1. So, the 1/4 1/2 1/4 ratio of tall, heterozygous and dwarf is mathematically condensable to the form of binomial expression ax plus by whole square that has the gametes bearing genes capital T or small t in equal frequency of half. The expression is expanded as given below. 1/2t plus 1/2t whole square is equals to 1/2t plus 1/2t into 1/2t plus 1/2t is equals to 1/4 capital T plus 1/2 capital T small t plus 1/4 small t. Mendel self-pollinated the F2 plants and found that dwarf F2 plants continue to generate dwarf plants in F3 and F4 generation. He concluded that the genotype of the dwarfs was homozygous small tt, What do you think he would have got had he self-pollinated a tall F2 plant? From the preceding paragraphs, it is clear that though the genotypic ratios can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait, it is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2 has T or T small t composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This he called a test cross. In a typical test cross, an organism, that is pea plant here, showing a dominant phenotype and whose genotype is to be determined, is crossed with a recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of the test organism. Figure 5.5 shows the result of typical test cross where violet color flower is dominant over white color flower. Using Punnett square, try to find out the nature of offspring of a test cross. What ratio did you get? Using the genotypes of this cross, can you give a general definition for a test cross? Based on his observation on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called the principles or laws of inheritance. The first law or law of dominance and the second law of segregation. 5.2.1 Law of dominance. Point 1. Characters are controlled by discrete units called factors. Point 2. Factors occur in pairs. Point 3. In a dissimilar pair of factors, one member of the pair dominant, the other is recessive. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion 3:1 obtained at the F2. 5.2.2 Law of segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of these is not seen at the F1 stage. Though the parent contains two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Of course, as a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. 5.2.2.1 Incomplete dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower, that is snapdragon or antirrhinum, is a good example to understand incomplete dominance. In a cross between true breeding red flowered and true breeding white colored plants, and the F1 was pink. When the F1 was self-pollinated, the F2 resulted in the following ratio, that is 1 red, 2 pink and 1 white. Here the genotype ratios are exactly as we would expect in any Mendelian monohybrid cross, but the phenotype ratio had changed from the 3:1, that is dominant, into recessive ratio. What happened was the R was not completely dominant over small r, and this made it possible to distinguish capital R small r as pink from capital R red and small r white. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand that every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, that is, as a pair of alleles. Now, these two alleles need not always be identical as in a heterozygote. One of them may be different due to some changes that it has undergone, about which you will read further on and in the next chapter, which modifies the information that particular allele contains. Let's take an example of gene that contains the information for producing an enzyme. Now there are two copies of these genes, the two allelic forms. Let us assume that the normal allele produces the normal enzyme that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for production of the normal less efficient enzyme, a non-functional enzyme, or no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele. That is, it will produce the same phenotype or trait that is the result in the transformation of substrate S. Such equivalent allele pairs are very common, but if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype trait will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represent the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. 5.2.2.2 Codominance. Till now, we were discussing crosses where the F1 resembled either of the two parents, that is dominance, or was in between incomplete dominance. But in the case of codominance, the F1 generation resembles both parents. A good example is different types of red blood cells that determine A-B-O blood grouping in human beings. A-B-O blood groups are controlled by the gene I. The plasma membrane of the red blood cell has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has three alleles, IA, IB and small I. The alleles IA and IB produce a slightly different form of the sugar, while allele small I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three capital I gene alleles, that is IA and IB are completely dominant over small I. In other words, when IA and small I are present, only IA expresses because small I does not produce any sugar. And when IB and I are present, IB expresses. But when IA and IB are present together, they both express their own type of sugar. This is because of codominance. Hence, red blood cells have both A and B type of sugars. Since there are 3 different alleles, there are 6 different combinations of these 3 alleles that are possible, and therefore a total 6 different genotypes of the human ABO blood types. How many phenotypes are possible? Table 5.2. Table showing the genetic basis of blood groups in human population. Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than 2, that is 3 alleles governing the same character. Since in any individual, only 2 alleles can be present. Multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has 2 alleles, capital B and small b. Starch is synthesized effectively by capital B homozygotes, and therefore large starch grains are produced. In contrast, small b homozygote have lesser efficiency in starch synthesis and produce a smaller starch grain. After maturation of the seeds, capital B seeds are round and the small b seeds are wrinkled. Heterozygotes produce round seeds, and so capital B seems to be the dominant allele, but the starch grains produced are of intermediate size in capital B seeds. So if starch grain size is considered as the phenotype, then from this angle the alleles show incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or the product, and that it has information for. It depends as much as on the gene product and the production of a particular phenotype from this product, and as it does on the particular phenotype that we choose to examine in case more than one phenotype is influenced by the same gene. 5.3 Inheritance of 2 genes. Mendel also worked with crossed pea plants that differed in 2 characters, as is seen in the cross between a pea plant that has seeds with a yellow color and round shape and one that had seeds of green color and wrinkled shape. Figure 5.7. Mendel found that the seeds resulting from the crossing of the parents had yellow colored and round shaped seeds. Here, can you tell which of the characters in the pair's yellow or green and round or wrinkled shape was dominant? Thus, yellow color was dominant over green and round shape dominant over wrinkled. These results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seeded plants. Let us use the genotypic symbols Y for dominant yellow seeds color and small y for recessive green seed color, capital R for round shaped seeds and small r for wrinkled seed shape. The genotype of the parents can then be written as capital R, capital Y, and small y, small y. The cross between the 2 plants can be written down as in figure 5.7, showing the genotypes of the parent plants. The gametes capital R, Y and small y unite on fertilization to produce the F1 hybrid, that is capital R, capital Y. When Mendel self-hybridized the F1 plants, he found that 3/4 of F2 plants had yellow seeds and 1/4 had green. The yellow and green color segregated in a 3:1 ratio. Round and wrinkled seed shape also segregated in a 3:1 ratio, just like in a monohybrid cross. 5.3.1 Law of Independent Assortment. In the dihybrid cross, as in figure 5.7, the phenotypes round yellow, wrinkled yellow, round green and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow, 1 green with 3 round, 1 wrinkled. This derivation can be written as follows: 3 round, 1 wrinkled, 3 yellow, 1 green, 9 round yellow, 3 wrinkled yellow, 3 round green and 1 wrinkled green. Based upon such observations on dihybrid cross, that is crosses between plants differing in 2 traits, Mendel proposed a second set of generalizations that we call Mendel's law of independent assortment. The law states that when 2 pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of characters. The Punnett square can be effectively used to understand the independent segregation of the 2 pairs of genes during meiosis and the production of eggs and pollen in the F1 capital R, capital Y plant. Consider the segregation of one pair of genes capital R and small r. 50% of the gametes have the gene capital R and the other 50% have a small r. Now, besides each gamete having either capital R or small r, it should also have the allele capital Y or small y. The important thing to remember here is that segregation of 50% capital R and 50% small r is independent from the segregation of 50% capital Y and 50% of small y. Therefore, 50% of the small r bearing gametes has capital Y and the 50% has small y. Similarly, 50% of the capital R bearing gametes has capital Y and the other 50% has small y. Thus, there are 4 genotypes of gametes, 4 types of pollen and 4 types of eggs. The 4 types are capital R, capital R, small r, capital Y and small y, each with a frequency of 25% or 1/4 of the total gametes produced. When you write down the 4 types of eggs and pollens on the 2 sides of the Punnett square, it is very easy to derive the composition of the zygotes that give rise to F2 plants. 5.3.2 Chromosomal Theory of Inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons it remained unrecognized till 1900. Firstly, communication was not easy, as it is now. In those days, and his work could not be widely publicized. Secondly, his concept of gene or factors in Mendel's world as stable and discrete units that control the expression of traits of the pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variations seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that factors were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. In 1900, three scientists, De Vries, Correns, and von Teschemann, independently rediscovered Mendel's result on the inheritance of characters. Also by this time, due to advancement in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus that appeared to double and divide just before each cell division. These were called chromosomes. By 1902, the chromosome movement during meiosis had been worked out. Walter Sotten and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's law. Recall that you have studied the behavior of chromosome during mitosis and during meiosis. The important things to remember are that chromosomes as well as genes occur in pairs. The two alleles of a gene pair are located on homologous sites on homologous chromosomes. Table 5.3, a comparison between the behavior of chromosomes and gene. They both occur in pairs. They segregate at the time of gamete formation such that only one of each pair is transmitted to a gamete. Independent pairs segregate independently of each pair, but one pair segregates independently of another pair. Can you tell which of these columns A and B represent the chromosome and which represent the gene? How did you decide? During anaphase of meiosis first, the two chromosome pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosomes of four different colors in the left and right columns. In the left column, orange and green is segregating together, but in the right hand column, the orange chromosome is segregating with the red chromosome. You can easily understand this thing with your book. Sotten and Boveri argued that the pairing and separation of pair of chromosome would lead to the segregation of a pair of factors they carried. Sotten united the knowledge of chromosomal segregation with Mendelian principles and called it chromosomal theory of inheritance. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis for the variation that sexual reproduction produced. Morgan worked with the tiny fruit flies, Drosophila melanogaster, which were found very suitable for such studies. They could be grown on simply synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes. The male and female flies are easily distinguishable. Also, it has many types of hereditary variation that can be seen with low power microscopes. 5.3.3 Linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid crosses carried out by Mendel in peas. For example, Morgan hybridized yellow-bodied white-eyed females to brown-bodied red-eyed males and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other and the F2 ratio deviated very significantly from the 9:3:3:1 ratio. Morgan and his group knew that the genes were located on the X chromosome and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combination were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage to describe this physical association of genes on a chromosome and the term recombination to describe the generation of non-parental gene combinations. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked. They show very less recombination, while others very loosely linked, they show higher recombination. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing showed 37.2% recombination. His student Alfred Stuartwent used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of whole genomes, as was done in the case of human genome sequencing project described later. 5.4 Polygenic inheritance. Mendel's studies mainly described those traits that have distinct alternate forms such as flower color, which are either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans we don't just have tall or short people as two distinct alternatives but a whole range of possible heights. Such traits are generally controlled by 3 or more genes and are thus called as polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele, that is, the effect of each allele is additive. To understand this better, let us assume that 3 genes A, B, C control skin color in human with the dominant forms A, B and C responsible for dark skin color and the recessive forms small a, small b and small c for light skin color. The genotype with all the dominant alleles, capital A, B, B, C, C will have the darkest skin color, and that with all the recessive alleles, small a, B, B, C, C, will have the lightest skin color. As expected, the genotype with 3 dominant alleles and 3 recessive alleles will have an intermediate skin color. In this manner, the number of each type of alleles in the genotype would determine the darkness or lightness of the skin in any individual. 5.5 Pleiotropy. We have so far seen the effect of a gene on a single phenotype or trait. There are, however, instances where a single gene can exhibit multiple phenotypic expression. Such a gene is called a pleiotropic gene. The underlying mechanism of pleiotropy in most cases is the effect of a gene on metabolic pathway which contribute towards different phenotypes. An example of this is the disease phenylketonuria, which occurs in humans. The disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase, that is single gene mutation. This manifests itself through phenotypic expression characterized by mental retardation and a reduction in hair and skin pigmentation. 5.6 Sex determination. The mechanism of sex determination has always been a puzzle before the geneticist. The initial clue about the genetic or chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, the cytological observations made in a number of insects led to the development of the concept of genetic or chromosomal basis of sex determination. Hanking in 1891 could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas the other 50% sperm did not receive it. Hanking gave a name to this structure as the X body, but he could explain its significance. Further investigation by other scientists led to the conclusion that the X body of Hanking was in fact a chromosome, and that is why it was given the name X chromosome. It was also observed that in a large number of insects, the mechanism of sex determination is of the exotype. That is, all eggs bear an additional X chromosome besides the other chromosome. On the other hand, some of the sperms bear the X chromosome, whereas some do not. Eggs fertilized by sperm having an X chromosome become females, and those fertilized by sperms that do not have an X chromosome becomes male. Do you think the number of chromosomes in the male and female are equal? Due to the involvement of the X chromosome in the determination of sex, it was designated to be the sex chromosome, and the rest of the chromosome were named as autosome. Grasshopper is an example of exotype of sex determination in which the males have only one X chromosome besides the autosome, whereas females have a pair of X chromosomes. These observations led to the investigations of number of species to understand the mechanism of sex determination. In a number of other insects and mammals including man, XY type of sex determination is seen where both male and female have same number of chromosomes. Among the males, an X chromosome is present but its counterpart is distinctly smaller and called the Y chromosome. Females, however, have a pair of X chromosomes. Both males and females bear same number of autosomes, hence the males have autosome plus XY, while female have autosomes plus XX. In human beings and in Drosophila, the males have one X and one Y chromosome, whereas females have a pair of X chromosomes besides autosomes. In the above description, you have studied about two types of sex determining mechanism, that is XO type and XY type, but in both cases, males produces two different types of gametes. A. Either with or without X chromosome, or B. Some gametes with X chromosome and some with Y chromosome. Such types of sex determination mechanism is designated to be the example of male heterogamety. In some other organism, example birds, a different mechanism of sex determination is observed. In this case, the total number of chromosomes is same in both males and females, but the two different types of gametes in terms of the sex chromosome are produced by females, that is female heterogamety. In order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of female bird has been designated to be the Z and W chromosomes. In these organisms, the females have one Z and one W chromosome, whereas the males have a pair of Z chromosome besides the autosomes. 5.6.1 Sex determination in humans. It has already been mentioned that the sex determining mechanism in case of humans is XY type. Out of 23 pairs of chromosome present, 22 pairs are exactly same in both males and females. These are the autosomes. A pair of X chromosome are present in the female, whereas the presence of an XY chromosome are determinant of the male characteristic. During spermatogenesis among males, two types of gametes are produced. 50% of the total sperm produced carry the X chromosome and the rest 50% has Y chromosome besides the autosome. Females, however, produced only one type of ovum with one X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. In case the ovum fertilizes with a sperm carrying X chromosome, the zygote develops into a female XX, and the fertilization of ovum with Y chromosome carrying sperm results into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy there is always 50% probability of either a male or a female child. It is unfortunate that in our society, women are blamed for giving birth to female children and have been ostracized and ill-treated because of this false notion. 5.6.2 Sex determination in honeybee. The sex determination in honeybee is based on the number of sets of chromosome an individual receives. An offspring formed from the union of a sperm and an egg develops as a female, and an unfertilized egg develops as male by means of parthenogenesis. This means that the males have half the number of chromosomes than that of a female. The females are diploid having 32 chromosomes and males are haploid that is having 16 chromosomes. This is called as haploid-diploid sex determination system and has special characteristic features such as the males produce sperms by mitosis, they do not have father and thus cannot have sons, but have a grandfather and can have grandsons. How is the sex determination mechanism different in the bird? Is the sperm or the egg responsible for the sex of the chicks? 5.7 Mutation. Mutation is a phenomena which results in alternation of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomena that leads to variation in DNA. As you will learn in chapter 6, one DNA helix runs continuously from one end to the other in each chromatid in a highly supercoiled form. Therefore, loss, deletion or grain insertion, duplication of a segment of DNA results in alternation in chromosome. Since genes are known to be located on chromosomes, alternation in chromosomes result in abnormalities or aberration. Chromosomal aberrations are commonly observed in cancer cells. In addition to the above, mutation also arise due to change in a single base pair of DNA. This is known as point mutation. A classical example of such mutation is sickle cell anemia. Deletions and insertions of base pairs of DNA causes frame shift mutation. The mechanism of mutation is beyond the scope of this discussion. At this level, however, there are many chemical and physical factors that induce mutations. These are referred to as mutagens. UV radiation can cause mutations in organism. It is a mutagen. 5.8 Genetic disorders. 5.8.1 Pedigree analysis. The idea that disorders are inherited has been prevailing in the human society since long. This was based on the heritability of certain characteristic features in families. After the rediscovery of Mendel's work, the practice of analyzing inheritance pattern of traits in human beings began. Since it is evident that control crosses that can be performed in a pea plant or some other organism are not possible in case of human beings, study of the family history about inheritance of a particular trait provides an alternative. Such an analysis of traits in several generations of a family is called the pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality or disease. Some of the important standard symbols used in the pedigree analysis have been shown in figure 5.13. As you studied in this chapter, each and every feature in any organism is controlled by one or the other gene located on the DNA present. In the chromosome, DNA is the carrier of genetic information. It is hence transmitted from one generation to the other generation without any change or alternation. However, changes or alternation do take place occasionally. Such an alternation or change in the genetic material is referred to as mutation. A number of disorders in human beings have been found to be associated with the inheritance of changed or altered genes or chromosomes. 5.8.2 Mendelian disorders. Broadly, genetic disorder may be grouped into two categories, Mendelian disorders and chromosomal disorders. Mendelian disorders are mainly determined by alternation or mutation in the single gene. These disorders are transmitted to the offspring on the same lines as we have studied in the principle of inheritance. The pattern of inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. Most common and prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait may also be linked to the sex chromosome as in case of hemophilia. It is evident that the X-linked recessive traits show transmission from carrier female to male progeny. A representative pedigree is shown in figure 5.14 for dominant and recessive traits. Discuss with your teacher and design pedigrees for characters linked to both autosomes and sex chromosomes. Color blindness. It is a sex-linked recessive disorder due to defect in either red or green cone of eye resulting in failure to discriminate between red and green color. This defect is due to mutation in certain genes present in the X chromosome. It occurs in about 8% of males and only about 0.4% of females. This is because the genes that lead to red-green color blindness are on the X chromosome. Males have only one X chromosome and females have two. The son of a woman who carries the gene has a 50% chance of being colorblind. The mother is not herself colorblind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. A daughter will not normally be colorblind unless her mother is a carrier and her father is colorblind. Hemophilia. This sex-linked recessive disease which shows its transmission from unaffected carrier female to some of the male progeny has been widely studied. In this disease, a single protein that is a part of the cascade of proteins involved in the clotting of blood is affected. Due to this is an affected individual, a simple cut will result in non-stopping bleeding. The heterozygous female that is carrier for hemophilia may be transmitted the diseases to son. The possibility of female becoming a hemophilic is extremely rare because mother of such a female has to be at least carrier and the father should be hemophilic, unviable in the later stage of life. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. Sickle cell anemia. This is an autosome-linked recessive trait that can be transmitted from parents to the offspring. When both the partners are carrier for the gene or heterozygous, the disease is controlled by a single pair of allele. hemoglobin A and hemoglobin S, HbA and HbS. Out of the 3 possible genotypes, only homozygous individual HbS show the diseased phenotype. Heterozygous HbA/HbS individuals appear apparently unaffected, but they are carrier of the disease, as there is 50% probability of the transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. The defect is caused by the substitution of glutamic acid. Glutamic acid by valine at the 6th position of the beta-globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein results due to the single base substitution at the 6th codon of the beta-globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the chain in the shape of the RBC from biconcave disc to elongated sickle cell-like structure. Phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait. The affected individuals lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of these in brain results in mental retardation. These are also excreted through urine because of its poor absorption of kidney. Thalassemia. This is also an autosome-linked recessive blood disease transmitted from parents to the offspring. When both the partners are unaffected carrier for the gene or heterozygous, the defect could be due to either mutation or deletion, which ultimately results in reduced rate of synthesis of one of the globin chain, alpha and beta chains, that make up hemoglobin. This causes the formation of abnormal hemoglobin molecules, resulting into anemia, which is characteristic of the disease. Thalassemia can be classified according to which chain of hemoglobin molecule is affected. In alpha thalassemia, production of alpha globin chain is affected, while in beta thalassemia, production of beta globin chain is affected. Alpha thalassemia is controlled by 2 closely linked genes, HbA1 and HbA2, on chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of the 4 genes. The more genes affected, the less alpha globin molecules produced, while beta thalassemia is controlled by a single gene, HbB, on chromosome 11 of each parent and occurs due to mutation of one or both the genes. Thalassemia differs from sickle cell anemia in that the former is a quantitative problem of synthesizing too few globin molecules, while the latter is a qualitative problem of synthesizing an incorrectly functioning globin. 5.8.3 Chromosomal disorder. The chromosomal disorders, on the other hand, are caused due to absence or excess of abnormal arrangement of one or more chromosomes. Failure of segregation of chromatids during cell division cycle results in the gain or loss of chromosome called aneuploidy. For example, Down syndrome results in the gain of extra copy of chromosome 21. Similarly, Turner syndrome results due to loss of an X chromosome in human females. Failure of cytokinesis after telophase stage of cell division result in an increase in a whole set of chromosome in an organism, and this phenomena is known as polyploidy. This condition is often seen in plants. The total number of chromosome in a normal human cell is 46, that is 23 pairs. Out of these, 22 pairs are autosomes and 1 pair of chromosome are sex chromosome. Sometimes, though rarely, either an additional copy of chromosome may be included in an individual, or an individual may lack one of any one pair of chromosome. These situations are known as trisomy or monosomy of a chromosome. Respectively, such a situation leads to a very serious consequences in the individual. Down syndrome, Turner syndrome, Klinefelter syndrome are common examples of the chromosomal disorders. Down syndrome. The cause of this genetic disorder is the presence of an additional copy of the chromosome number 21, that is trisomy of 21. This disorder was first described by Lang John Down in 1866. The affected individual is short-statured with a small round head, furrowed tongue and partially open mouth. Palm is broad with characteristic palm crease. Physical, psychomotor and mental development is retarded. Klinefelter syndrome, the genetic disorder, is also caused due to the presence of an additional copy of X chromosome resulting into a karyotype of 47, XXY. Such an individual has overall masculine development. However, the feminine development of breast, that is gynecomastia, is also expressed. Such individuals are sterile. Turner syndrome, such a disorder is caused due to the absence of one of the X chromosome, that is 45 with XO. Such females are sterile as ovaries are rudimentary besides other features including lack of other secondary sexual character. Summary. Genetic is a branch of the biology which deals with the principles of inheritance and its practices. Progeny resembling the parents in morphological and physiological features has attracted the attention of many biologists. Mendel was the first to study this phenomena systematically. While studying the pattern of inheritance in pea plants of contrasting characters, Mendel proposed the principles of inheritance which are today referred to as Mendel's laws of inheritance. He proposed that the factors, later named as genes, regulating the characters are found in pairs known as alleles. He observed that the expression of the characters in the offspring follow a definite pattern in different first generation F1, second F2 and so on. Some characters are dominant over others. The dominant characters are expressed when factors are in heterozygous condition, that is law of dominance. The recessive characters are only expressed in only homozygous conditions. The characters never blend in heterozygous condition. A recessive character that was not expressed in heterozygous condition may be expressed again when it becomes homozygous. Hence, characters segregate while formation of gamete, that is law of segregation. Not all characters show true dominance, some characters show incomplete and some show co-dominance. When Mendel studied the inheritance of two characters together, it was found that the factors independently assort and combine in all permutations and combination, that is law of independent assortment. Different combinations of gametes are theoretically represented in a square tabular form known as Punnett square. The factor now known as gene on chromosome regulating the characters are called the genotype, and the physical expression of the character is called phenotype. After knowing that the genes are located on the chromosome, a good correlation was drawn between Mendel's law, that is segregation and assortment of chromosome during meiosis. The Mendel's law were extended in the form of chromosomal theory of inheritance. Later, it was found that Mendel's law of independent assortment does not hold true for the genes that were located on the same chromosome. These genes were called as linked genes. Closely located genes assorted together and distantly located genes due to recombination assorted independently. Linkage maps therefore correspond to the arrangement of genes on a chromosome. Many genes were linked to sexes also and called as sex-linked genes. The two sexes, male and female, were found to have a set of chromosomes which were common and another set which was different. The chromosomes which were different in two sexes were named as sex chromosomes. The remaining set was named as autosome. In humans, a normal female has 22 pairs of autosomes and a pair of sex chromosome XX. A male has 22 pairs of autosomes and a pair of sex chromosome as XY. In chickens, sex chromosomes in male are ZZ and in female are ZW. Mutation and is defined as a change in the genetic material. A point mutation is a change of single base pair in DNA. Sickle cell anemia is caused due to change of one base in the gene coding for beta chain of hemoglobin. Inheritable mutation can be studied by generating a pedigree of family. Some mutation involve changes in whole set of chromosomes, that is polyploidy, or change in a subset of chromosome number aneuploidy. This helped in understanding the mutational basis of genetic disorders. Down syndrome is due to trisomy of chromosome 21, where there is an extra copy of chromosome 21 and consequently the total number of chromosomes become 47. In Turner syndrome, and one X chromosome is missing and the sex chromosome is as XO, and in Klinefelter syndrome the condition is XXY. These can be easily studied by analysis of karyotypes.